Welcome to this talk on white blood cells. And just before we get into the fascinating area of the white blood cells themselves, let's just give an overview by looking at the nature of the blood. Now, as you probably know, blood consists of plasma and cells. And the plasma is the fluid part in which the cells float. So there are various types of blood cells floating in the plasma. The plasma is the liquid component. So what does plasma contain? Well, it's largely water. But there's also things dissolved in this water to form the plasma, such as the plasma proteins, particularly albumin, which is a large plasma protein. And there's also waste substances. For example, urea is the body's main way of excreting nitrogen waste products. And urea is created from nasty, toxic, nitrogen-containing components that the liver takes out of the blood. And the liver takes these nasty, poisonous components and converts them, or rather incorporates them, into urea. So the liver produces urea. This urea circulates in the blood, and it's excreted by the kidneys. So the urea is carried in the plasma as well. And there's nutrients carried in the plasma, of course, from the digestive tract to various parts of the body, or really to all parts of the body where nutrients are used. So, for example, there's glucose in the blood. There has to be glucose in the blood, otherwise you suffer from a condition called hypoglycemia. There's also amino acids in the blood, the building blocks of proteins. And there's all the other nutrients as well circulating around in the blood, like minerals and vitamins. The minerals can be in the form of electrolytes as well. For example, there's Na+, which is sodium, there's K+, which is potassium, there is Cl-, which is the chloride ion, and there's calcium ions, that's the Ca2+ ion. And there's also hormones circulating around in the plasma, from endocrine glands where they are produced to target organs where they give a signal. The endocrine hormones are giving signals to various target tissues around the body. So that's the fluid part, that's the plasma. And by far and away the most abundant type of cell floating in the plasma are the red blood cells, the erythrocytes. Erythro means red and cyte means cell. And these erythrocytes are biconcaved discs. They're about 7 micrometers in diameter, and they have an area of central pallor, which is equal to 1/3 or less of the total diameter of the red cell. And the area of central pallor is a paler coloured area because the red cells are biconcaved discs and they're thinner in the middle. This is good because it allows them to be flexible and squash down when they go through small capillaries. And the red cells are red because they carry a pigment called hemoglobin. And each hemoglobin molecule is able to carry 4 molecules of oxygen from the lungs around in the circulatory system to the tissues where that oxygen is essential and needed for all metabolic processes, because the oxygen is used to produce energy. So when the hemoglobin is oxygenated, we call it oxyhemoglobin. When it gives up its hemoglobin, it becomes reduced hemoglobin, which we call deoxygenated hemoglobin. So this is going on all the time. But as well as the red cells, there are white cells in the plasma. Now the white cells are called leukocytes. The leukocytes are the white cells. And if there's too many white cells, or not too many really, but if there's an increase in the number in white cells, we call that a leukocytosis. A leukocytosis. So what might give rise to a leukocytosis? Well, an increased number of white cells is normal if there is an infection. Because if there's an infection, it's the white blood cells that fight this infection, get rid of it and kill it off. Because if they didn't do that, the infection will multiply in the body and in the tissues and in the blood, and even a very small infection could kill us. It could be absolutely fatal if the white blood cells are not combating the infection. So if we look at the blood, There's a blood test we can do called a differential white cell count. If we look at that, we see there's an increase in the number of leukocytes. There's a leukocytosis. Then we think, leukocytosis. I strongly now suspect this patient has got some sort of infection somewhere, because that's what white blood cells do. They fight infection. Absolutely vital. Now, the converse of a leukocytosis is a leukopenia. Penia means not enough. And if there's not enough white blood cells, this makes the individual with the leukopenia very prone to infection, sometimes overwhelming, fatal infection. So it's very important to have adequate numbers of white blood cells in the blood at any one time. So we have the white cells, which are the leukocytes. Now, there's two main classifications of leukocytes we're going to talk about today. The first classification are called the granulocytes, and the other classification we'll talk about in a minute are called the agranulocytes. But first of all, let's talk about the granulocytes. Now, why are the granulocytes so called? Well, these are white blood cells that have granules in their cytoplasm. When people first looked down light microscopes at these leukocytes, they said, oh, look, those ones have got granules in the cytoplasm. Let's call them granular cells or granulocytes. But in actual fact, today we know that they're not really granules. They're not little bits of grit in the cytoplasm. What they were actually seeing were vesicles, which are membrane-bound areas within the cytoplasm of the cells. And these vesicles actually contain various important chemicals that allows the cells to carry out their important defense functions. For example, some of the vesicles contain lysozyme, which is a type of digestive enzyme which can digest bacteria. And they also carry inflammatory mediators such as histamine in other types of cells, in some types of granulocytes as well. So the granules are actually vesicles containing important chemicals. So white cells can be granulocytes or agranulocytes. We're now thinking about the granulocytes. And one type of granulocyte is called a basophil. Now, you virtually never see basophils in the blood, because what the basophils do is they migrate out of the blood into the tissues. And in the tissues, they sit around in the tissues as mast cells. And mast cells are present in the tissue to initiate or to bring about an inflammatory response. So when the tissue is injured or insulted, mast cells will degranulate, and that will release histamine into the tissues. And the histamine is an inflammatory mediator. So the granules inside the mast cells contain histamine, and this histamine is released into the tissues, and it brings about an inflammatory response. But the basophils must be present in the blood in order to migrate into the tissue spaces. So mast cells derive from basophils. If they're in the blood, we call them basophils. If they're in the tissues, we call them mast cells. And then there's another type of granulocyte called an eosinophil. Now, eosinophils, like basophils, have a two-lobed nucleus, and they also contain these granules, which we now know are actually vesicles. Now, if there's an increase in the number of eosinophils, that's called an eosinophilia. So an eosinophilia is an increase in the number of eosinophils. Now, if someone's got an eosinophilia, do you know what that will indicate? So you've got a patient, might not feel too well. You take blood, you do a differential white cell count, you find there's lots of eosinophils. That patient has an eosinophilia. What does that indicate? Well, it could indicate parasitic infections with large parasites such as intestinal worms or worms in the tissues, tissue worms, because eosinophils will help to combat worm infections and other multicellular organisms which can cause infection, the parasitic infections. So if you've got parasitic infections, more eosinophils are released to try and combat that infection. So you could think, especially if you're in the tropics, you could think, this patient's got an eosinophilia. Ooh, I wonder if that means he's got a parasitic infection of some sort, and then you could then look for that parasitic infection and treat it. But it can also indicate allergic state. So you can get an eosinophilia in some allergic reactions as well. And it can also indicate chronic inflammation, eosinophilia. So granulocytes can be basophils or eosinophils, and granulocytes can also be neutrophils. And neutrophils contain 3 or 4 lobes in their nucleus, so they're sometimes called polymorphonucleocytes. Poly means many, morph means shape, nuclear means the nucleus, and cytes means cells. So they're polymorphonucleocytes. And again, these contain granules in their cytoplasm, which we now know are vesicles, albeit rather small granules. And there's actually a reserve of neutrophils stored in the bone marrow, and if there's an infection, these neutrophils will be released in large numbers into the circulation so they can combat the infection. And in fact, the neutrophils are the most abundant form of granulocytes by quite a long way. You don't get basophils much in the blood, and you don't get many eosinophils normally, but you do get quite a few neutrophils. So any bacterial infection, particularly bacterial infection, and lots of these very useful neutrophils will be released into the blood to combat it. And they migrate out of the blood vessels into areas of infected tissue as well. So if there's bacterial infection in a tissue, you'll get lots of neutrophils migrating out of the blood into the infected tissue, and the neutrophils will start to phagocytose the infection. They will start to ingest and digest the infecting bacteria. And if you get an increase in the number of neutrophils, we call that a neutrophilia. So if you looked at a differential white cell blood count, you found an increase in the number of neutrophils, particularly the patient had a neutrophilia, then you would suspect that that patient had a bacterial infection, and you could investigate and treat them accordingly. Now, if there's not enough neutrophils, that would be called a neutropenia, and that patient is going to be prone to infection. And if there's a very significant reduction in the number of neutrophils present in the circulation, we will call that an agranulocytosis. A means without, so that would mean without granulocytes. Agranulocytosis, and that would be a very dangerous situation for a patient to be in. So white cells can be granulocytes, but white cells can also be agranulocytes. Agranulocytes literally means cells without granules. Now this isn't actually true. We know that these cells do often contain vesicles, but they're less obvious on light microscopy. So they got the name agranulocytes, and that's what we're stuck with. So what sort of agranulocytes might be present in the blood? Well, there's cells called monocytes. Now monocytes contain a kidney bean or a kidney-shaped nucleus, and they circulate around in the blood, and they are phagocytic. But monocytes can migrate from the capillaries into the tissue spaces. And when the monocytes are in the tissue spaces, they change their name. They're no longer called monocytes, but we call them macrophages. So when monocytes migrate into the tissue, they change their name and become macrophages. It's a bit like the basophils. Remember when they moved into the tissue, they changed the name from basophils to mast cells. Well, it's the same with monocytes. In the blood, they're monocytes. In the tissues, we call them macrophages. And macro means big, and phage means to eat. So the macrophages are big eaters. They will eat bacteria. They will eat clumped together viruses. They will eat dead tissue cells. And when they eat all this stuff, they get bigger and bigger, and they become great big cells called macrophages. Now, sometimes monocytes migrate from the blood into the tissue spaces, and they differentiate into another type of cell altogether called a dendritic cell. Now, a dendritic cell looks a bit like an octopus. It's kind of got little legs going off it called dendrites. And what these dendrites do is they collect antigens, an antigen being any foreign material, any foreign thing like a virus or something like that. Something infecting the individual that shouldn't be there. And once the dendritic cell is activated by the presence of an antigen, it will migrate in the lymphatics to a lymph node. And in the lymph node, it will present this antigen to the lymphocytes, which will then mount an immune response. So we call the dendritic cells antigen presenting cells because they present antigens and they can then mount an immunological response to the antigen which has caused the infection. Now, thrombocytes are the platelets. They're circulating around in the blood and they're absolutely vital for the process of blood clotting, and we can classify those also as a type of agranulocyte. But the most abundant agranulocytes in the blood are the lymphocytes. As well as spending time in the blood, the lymphocytes spend time in the lymphatic system, indeed, as the name implies. And there's two types of lymphocytes. There's the large lymphocyte and there's the small lymphocyte. Now, the large lymphocyte have a smaller nucleus than the smaller lymphocytes comparatively, and they have granules in their cytoplasm because these are the natural killer cells, the so-called NK cells. So the large lymphocytes are the natural killer cells, and they will kill cells which are infected by a virus. So if a virus is growing inside a cell, then bits of that virus will come to the surface of the body cell in which the virus is growing. These antigenic molecules on the surface of the infected body cell will be recognized by the natural killer cell. The natural killer cell will then drop chemicals onto the surface of the cell which will kill the cell, and it will kill the body cell. Now, what this means is natural killer cells are actually going around killing some of the body's own cells. But remember, these are body cells which are infected by viruses. And if the cells weren't killed, the viruses would be released and infect many more cells. So what the natural killer cells are doing is they're not messing about trying to kill individual viruses. They just kill the whole infected body cell, but in the process kill the virus as well and stop the virus from spreading. So the lymphocytes can be large, which are the NK cells, but lymphocytes can also be small. Now, if there's an increased number of lymphocytes, we call this a lymphocytosis. So an increased number of lymphocytes is a lymphocytosis. So you do a differential white cell count, there's an increased number of lymphocytes, there's a lymphocytosis, and that should make you think, a lymphocytosis. Now, it's lymphocytes that combat viral infections, so the number of lymphocytes will increase in viral infections. So as a lymphocytosis, you should think, a viral infection, and then you can investigate and treat the patient accordingly for the viral infection. So lymphocytes can be large or small. The small lymphocytes can be divided into B or T. Now, the B lymphocytes mature in the bone, and the B lymphocytes will differentiate into a group of cells called plasma cells, and it's the plasma cells which produce the antibodies. So really what we're saying is it's the B cell lines that produce the antibodies. The antibodies are the immunoglobulins, the immune proteins that get rid of infecting antigens, bacteria or viruses. It is the B lymphocytes that produce the antibodies that mount the specific acquired immune response. But as well as being B-type small lymphocytes, there's also T small lymphocytes. And the T small lymphocytes, they mature in the thymus gland. And there's three types of T small lymphocyte: T helpers, T suppressors, and T cytotoxic. Now, the T helper cells stimulate various cells in the immune system to get them going, to make them active when there is an infection. And in particular, the T helper cells stimulate the B cells, that is, the small T lymphocytes stimulate the small B lymphocytes to tell them to produce the antibodies. And actually, if someone's infected with HIV, the human immunodeficiency virus, that can cause acquired immunodeficiency syndrome because the HIV virus kills off the T helper cells. If the T helper cells are killed off, that means there's no cells to stimulate the B cells to produce the antibodies. But as well as that, when it's time to finish mounting an immune response, the B cells need to be suppressed. So there's T suppressor cells as well, which limit the amount of immune response that the B cells are mounting. So the T helper cells stimulate the B cells to produce antibodies. The T suppressor cells tell it when they've made enough that the infection's got rid of and we can stop mounting this immune response now. And the third type of small T lymphocyte is the T cytotoxic cell. And again, these cytotoxic cells, well, cyto means cell, toxic means poisonous. So these cytotoxic cells will kill virally infected cells in a similar way to the natural killer cells, but the T cytotoxic cells will kill virally infected cells in a more specific way. They're acting against a more specific type of virus. And actually, it's interesting, the T cytotoxic cells and the NK cells, as well as killing virally infected cells, can also kill cancer cells as well. So a good immune system is vital for keeping cancers at bay. So we've said the white cells can be granulocytes or agranulocytes. We've said the granulocytes can be basophils, eosinophils or neutrophils. We've said that the white cells can be agranulocytes. The agranulocytes can be monocytes, and the monocytes give rise to macrophages or dendritic cells. We've said agranulocytes can be thrombocytes. We've said agranulocytes can be lymphocytes. Lymphocytes can be large or small. The large lymphocytes are the natural killer cells. The small lymphocytes can be B or T, and the T can be T helpers, T suppressors, or T cytotoxic cells. So that was a brief introduction into the types of white blood cells in the blood and their absolutely vital role in immunity.